body, the physical substance of the human organism, composed of extracellular materials and organized into systems, organs, tissues, and living cell, the basic structural function and biological unit of all known organism, the smallest unit of life, often called the building blocks of life. In the cell, we found genes, the basic functional unit of heredity, that is found in the tiny spaghetti-like structures called chromosomes. Our body is made up of billions of cells. That is why scientists have taken interest in the small details in our body to protect us from the diseases such as single gene defects.
Have you heard of disease caused by single gene defects? Cancer, heart disease, diabetes, hemophilia, and AIDS are some examples of it. Scientists have been working for decades on ways to cure disorders caused by a mutation in genes. And they come up with gene therapy.
Gene therapy is a technique that modifies a person's gene to treat or cure diseases. It works in several mechanisms, replacing a disease-causing gene with a healthy copy of the gene. But before anything else, let us know first the basics.
So what is a gene? Genes are the basic unit of heredity. They contain information for our individual characteristics, like eye and hair color. These are a section of DNA that oversee different functions, which includes making of proteins. Genes come from each of our parents, wherein we may inherit their physical traits, as well as mutation in genes that can sometimes cause diseases and health conditions. That's when gene therapy comes in.
So how does gene therapy work? Gene therapy replaces a faulty gene or adds a new one to cure and improve your body's ability to fight diseases. In order to insert new genes directly into cells, scientists use a vehicle called vector, which is genetically engineered to deliver the gene. Certain viruses are used as vectors since they can easily deliver the new gene by infecting the cell. But modified viruses don't cause diseases. Gene therapy can be used to modify cells inside or outside the body. These methods are in vivo and ex vivo. In vivo gene therapy is done inside the body. A vector will be injected carrying the gene directly into the part of the body that has defective cells. While ex vivo is done outside of the body, cells are taken out and the vector containing the desired gene is introduced into these cells. The cells are left to multiply in the laboratory and are then injected back into the patient, where they continue to multiply and eventually produce the desired effect.
Although gene therapy is a promising treatment option for several diseases, this technique remains risky and is still under study. To make sure that it will be safe and effective, unwanted immune system reaction might happen. The body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and, in severe cases, organ failure. Vectors might as well target the wrong cells. It is possible that the altered viruses may affect additional cells, not just the targeted cells containing mutated genes.
Despite of some possible risks, this treatment option is potentially useful for some inherited disorders, cancers, and viral infections. This technique has the ability to cure many of the diseases that have affected our society for years. In the coming future, gene therapy will play an important part in many people's lives. The role of doctors and patients will be changing in the coming years, and it will all be due to this therapy.